Machine learning and electron microscopy image analysis, particles identification. Next slide. Okay, so our first topic is the big picture of machine learning and electron microscopy. And in general, electron microscopy is a technique that uses a beam of electrons to create highly detailed images of small objects, and it provides much higher magnification and resolution than regular light microscopes. And machine learning algorithms have been used to identify which data points in images deserve greater focus. And in the conversation of the consideration of automated experiments, there are three components to consider with complete automation. So that is the engineering controls, which allow the operation of the microscope through external electronics, relying on manufacturer programmed interfaces. And secondly, we have machine learning algorithms, which generate a sequence of operations for the computer. And finally, the perceived goal and target is a list of potential outcomes based on the experiment, which guides the process. And the overall goal of this is to minimize human intervention because humans are prone to making errors. Next slide. So now we're going to be talking about image processing versus machine learning in scanning electron microscopy. So like I mentioned earlier, this is useful for analyzing smaller materials, but processing scanning electron microscopy images is hard. So traditional methods like edge detection struggle with images that are really complex or dense. So machine learning, especially convolutional neural networks and you only look once models, can automatically detect nanoparticles by learning from large data sets. And so machine learning is more accurate in general, but it requires more labeled data and obviously powerful computers. And so combining traditional and machine learning methods can help improve detection in general. Next slide. So now the different models that are used to classify the images. So first off, we have convolutional neural networks, which also include AlexNet and visual geometry group networks. So these are deep learning models that are used for image recognition, detecting different patterns like edges and textures. And so AlexNet was one of the first successful models. And then the visual geometry group network is really simple and effective, and it's great for transfer learning. But overall, all of these require a lot of memory and computing power, so that could be a challenge. Inception and residual network next is, so these improve object recognition by using different filter sizes to capture both small and large details. But because it's so complicated, it makes it harder to modify. And it requires a lot of computational resources and training data. And then lastly, convolutional neural networks and you only look once models are, they refine object detection by segmenting and detecting overlapping objects that achieve higher accuracy, but overall at a slower pace and higher computational requirements. Next slide. So now, why do we want to precisely image nanoparticles? So nanoparticles are really small. They're less than 100 nanometers, and they have unique properties. And they're really useful in medicine, electronics, material science, and they're useful in many different fields, such as biochemistry, semiconductors, and nanotechnology advancements with drug delivery, which is something that is being worked on in the biochem lab. And the imaging of these nanoparticles can help reveal different properties that are useful for many different applications. Next slide. So now our short-term and long-term goals for this project. Our short-term goal is to identify and count spherical and cubical nanoparticles on the scanning electron images and report if there's any alignment or ordering present in these images. And our long-term goal is to count and identify these arbitrary particles and on optical and scanning electron microscope images and report the different types of alignment, patterning, and short range or long range of these parameters.
So for preparing our data sets, we are planning to start with dividing our data by using the 70-30 ratio, one of the industry standards, for our in-sample/training data and our out-of-sample/validation data. For our training data, we plan to use proportions of training data of growing magnitude, such as 10, 100, 1000, or by a factor of 10. We are labeling/tagging our training data for machine learning by using image processing. And we are going to train the model on different subsets of data by testing and recording their performance on the same training data. And then finally, we will plot the results. So for our data sets and code, our code includes parts that are ChatGPT generated, uses CV libraries and Stack Overflow. For our images, we have 2,400 SEM images divided into 3 different data sets of spherical particles, with one that is disordered, with an example being in the top right, and the other that is ordered, with the example being in the bottom right, and cubical particles, with an example being the middle image. Additionally, we have 400 optical microscope images of silicon carbide particles, with an example being the most bottom left image. And so we first started by doing image processing on our optical images. So the top image shows the folder path of 16 optical microscope images and a couple SEM images the code was initially run on. The bottom left image shows an example of one of the initial results for the optical microscope image, while the 2 right photos show the original image and the image after background subtraction had been applied. We stopped using the optical microscope images because of its arbitrary shape and difficulty with focus. With the optical microscope image, it would have required deconstructing them into regular shapes and then summarizing or combining them. So we therefore decided to start off with regular shapes and then come back to the optical microscope images after mastering the spherical and cuboidal images. So for our training, we are using the 3 different data sets. In the spherical disorder dataset, some notable features include how there are clusters of group of particles. However, they are in a scattered manner, making them disordered, as shown in the top left image. There's also sections of different elevation, as shown in the top middle image. And also there's different 3D formations, as shown in the top right. For the spherical order dataset, there are also clusters, but in an ordered manner, as shown in the middle left image. And also similar to the disordered dataset, there is a difference of elevation between certain parts. And there's also a difference in contrast between the background and the particle for different section of the image, as shown in the middle right. And then finally, for the cuboidal images, they are scattered in a disordered format, but also include other shapes as indicated by the long rectangle in the bottom middle image, and there are also large clusters of particles as shown in the bottom right. So on the image to the left, there are 217 particles in the image, and so with a series of 5 trials, with the number of particles incorrectly identified, we use that to construct a learning curve. So for trial 2, shown in the top corner, it failed to correctly identify 129 different particles, failing to distinguish between individual particles and clusters of particles, which compared to trial 5, shown in the bottom left corner, which incorrectly identified 24 particles. It shows an improvement as it was much better at distinguishing individual particles in a group cluster. So in the different data sets, we got varying results for different shaped particles, as the cuboidal shaped particles had better results than the spherical particles. Additionally, across each dataset, there were varying results. As for the spherical order dataset, certain images struggled with identifying the presence of particles, and it would also often identify parts of the scale bar. The spherical order dataset, however, was good at identifying the particles in their ordered alignment, as the circles were often aligned in a straight or slightly curved lines. For the spherical disorder dataset, it was good at identifying a majority of the particles, but struggled with tin carbon and the images of different photography and 3D images. For the cuboidal images particles, it was good at identifying particles of medium size, but struggled to identify the individual particles when there was overlap, and also the smaller particles.
So after we do all the training, we have to validate all our data sets. So essentially, what we do is we have a validation curve, which tells us how our model is improving over time. So on the y-axis, we have the percentage of incorrectly identified, missed, or misidentified particles. While on the x-axis, we have the amount of images processed through the model. So as we can see, the more we train our model, the more accurate it is in detecting particles in images. So take the images on the right as an example. The image on the bottom looks like it has identified lots and lots of particles. Meanwhile, the image on the top misses a bunch of particles, which we can obviously see. So this is because after training the data, after training the model with the data for a while, it's better at recognizing particles. So some common patterns we've noticed with our image detection is that we have a bunch of errors. One of our main patterns being the fact that clustered particles are often identified as one. Additionally, since some particles have overlap, the model also often considers them to be more than two. Along with that, we have an issue with overcounting and undercounting, which results from all the issues described earlier. And we in the cuboidal datasets, we have an issue with tilted particles not being recognized. Another back to the spherical particles, we have some images have pretty big issues with only the scale bar, parts of the scale bar being identified as particles, but not any of the actual particles being identified as particles. So we look forward to improving our code and making sure that particles are no longer misidentified or unidentified. As we mentioned earlier, one of our long-term goals was to be able to identify optical microscope images. However, this is a very tricky task, as oftentimes us humans are in conflict on which particles in and whether or not two particles in an image can be considered as one particle or split into separate particles. So if we want to do that, we have to define a standard for considering particles, while also we need to finish up and solve the issue of misidentifying particles in SEM images. We would like our code to count particles of all sizes of all shapes and sizes regardless of morphology, and be able to report alignment patterns long or short range order. So our ultimate goal for this project is to be able to incorporate an image detector into ASDRP scanning electron microscope to have it automatically take pictures for us. The problem with scanning electron microscopes right now is that it takes a very long time to take thousands and thousands of pictures, which is why it's necessary for a machine to do it for us. The issues mostly reside from the fact that the process is very, very repetitive. And removing the repetitive parts can save lots of time, energy, and money, which is very important for industry. Here are all our references. And we would like to thank our parents for the support they've given us and ASDRP for the opportunities they have given. And we would like to especially thank our advisors, Dr. Starostina and Professor Downing, for guiding us through this project. Do we have any questions?
Yes, I have a question. You had a scale where you showed the inaccuracies versus the number of particles being tested. Could you go back to that slide?
Sure.
Yes. Why are there no X and Y legends on the slide?
Oh, I put it on the side.
No, I mean, a scale. There's no scale.
Oh, because we haven't actually we haven't actually done the validation process yet. This is just what our ultimate goal.
Oh, so this is a hypothetical graph, not an actual graph of your results.
Yeah. Sorry for the confusion.
Okay, I'm done.
Okay, I have a question. You guys mentioned a lot in your second to last slide about some of the difficulties you had with determining the particles. What specifically are some steps you have taken to resolve those issues?
So I can speak for the cuboidal particles. So what I've done is I've tried adjusting the contour area requirement for a particle to be considered a particle, but that has worked with varying success. Some images do better than others when I adjust the contour area. And an issue that we've had with adjusting the contour area is that the lower we set it to be, uh, the more likely it is for the scale bar, for parts of the scale bar to be considered particles. Uh, Sophia and Anya, would you guys like to talk about spherical
Yeah, so
particles?
for like the spherical images, one thing like I've been working with is like trying background subtraction. But for different spherical images, originally I tried with the mean, using the mean as the background. But for some images, the mean value is actually the particle and not actually the background. So I've been trying with different methods to be able to correctly identify the background. And so that way, if you with identifying the background, it helps for, especially in the beginning, they would try to identify the background as a particle instead of actually the particle. And so that's one way been trying, and then also switching the order of steps and trying to see if that helps with it.
Do we have any more questions? Assume not.